In this video, you will learn about transcription. I will tell you about what is transcription, then what is the process of transcription. Takes place in 3 steps: initiation, elongation, and termination. But before that, I will tell you about the role of enzyme that is utilized for the process of transcription, that is RNA polymerases. I will tell you brief about the RNA polymerases, and also I will explain about the template strand. Let's start with what is transcription. Transcription is the process of converting DNA sequence into messenger RNA. So it is the process in which messenger RNA is formed by copying the DNA sequences. Transcription, or why we need to copy the DNA in the form of messenger RNA, because the information for the synthesis of protein are stored in the DNA in the form of genetic codes. So these codes are then copied in the form of messenger RNA, and this code is taken for the synthesis of the amino acids, and these amino acids sequence then combine together to form the proteins. The transcription process basically takes place to carry the code, the genetic code from the DNA towards the protein synthesis for the desired or the specific protein synthesis. How this messenger RNA copies the information from the DNA by the process of transcription. To understand it easily, basically the transcription process is divided into 3 steps. First step is initiation step, from where the transcription basically begins, then proceeded by elongation, and then finally there is the termination. By these 3 steps, the synthesis of messenger RNA takes place. Before going into detail into the steps, each step of the transcription, the most important thing that is required for the synthesis of messenger RNA, as you can see here in this diagram, the RNA polymerase. The RNA polymerase is the enzyme which catalyzes the synthesis of messenger RNA. Let's see first of all what is this RNA polymerase and how it catalyzes the synthesis of messenger RNA. As there are 3 different types of RNA: messenger RNA, ribosomal RNA, and transfer RNA. In prokaryotes, the single RNA polymerases, this one enzyme, catalyzes all these 3 different types of RNAs, while in case of eukaryotes, there are 3 different types of RNA polymerases: RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. That we will study in detail when we will study about the transcription in prokaryotes and transcription in eukaryotes. Now let's see about the process of transcription, that how the process of transcription takes place. Then before that, I told you we have to see what this RNA polymerase is and how it catalyzes the synthesis of RNA. RNA polymerase is the enzyme that catalyzes the transcription by synthesizing RNA from the DNA template. So DNA is the double helical structure that is wrapped around tightly in a helical shape, as you can see here in this. The template strand of DNA to which complementary base pairing will be attached. This complementary base pairing will be attached through hydrogen bonding. By copying these sequences, the RNA transcript will be formed. So the DNA has 2 strands. Which strand will be used as a template strand? So the one strand that is from 3' to 5' is known as the antisense strand, and the other strand, which is 5' to 3', is known as the sense strand. As the RNA transcript that will be formed will always be from 5' to 3'. So it will copy the sequences of the strand which is 3' to 5', which is known as antisense strand. So similarly, this strand will be formed 5' to 3'. And these sequences are similar to the sequences, they match the sequences that are on the opposite strand known as the sense strand. So the sense strand sequences will be similar to the RNA transcript sequences. So the sense strand basically contains the information that will be used for the synthesis of the desired and specific protein. So this code is basically copied and is carried to the cytoplasm and is carried to the ribosomes for the synthesis of the for the combination of the amino acids for the synthesis of the specific and desired protein. So which strand will be used as a template strand? The antisense strand, which is 3' to 5', that will be copied, and the messenger RNA formed is 5' to 3', which will having the sequences similar to the sense strand. Now let's start. This was about the RNA polymerases, that basically what functions are performed by the RNA polymerase and which strand it's going to copy the sequences and what is the template strand. Now let's start with the steps of the transcription one by one. Let's start with the first step, which is the initiation step. Initiation is the step in which the RNA polymerase enzyme binds to the DNA. As the DNA is along sequences, to which region these RNA polymerases will bind. So for binding of the RNA polymerases, there is a specific sequences which is known as promoter. So the promoter are the nucleotide bases or sequences to which this RNA polymerase will bind and will initiate the process of transcription. And before initiation, there are some sequences which we can also call them pre-initiation sequences. So promoter, it contains the resequences to which this machinery or the transcription machinery will basically binds, and then this is basically the coding region, then followed by the coding region which will be copied by messenger. As you can see here, this is the template strand which is copied by the messenger RNA, and this is the sense strand. But from where the transcription has to begin, how would this RNA polymerases will know? RNA polymerase will bind to this promoter region, also act as a regulatory region. When the transcription is required, it will bind to this promoter region, and when the transcription is not required, this regulatory region will turn off. So these are the promoter sequence. The promoter sequences, what are the sequences basically which will be recognized by the RNA polymerases for transcription? These sequences are Tata box. Tata box is present -10. These are the sequences. This is the promoter region. Most commonly we say that this is the upstream region, and then this is the downstream region. This is basically the site from where the transcription starts. So this is the initiation, start of transcription. This is the initiation step. We call them +1. So from 1+1, basically this is the region to which the RNA polymerase will bind, and then 5' untranslated region. This is the basically the coding sequences region, and then 3' untranslated region. From this initiation start site, the upstream, there are sequences that varies in different organisms. It may vary from, these sequences may vary from 100 to 1000 sequences. The most important Tata box is in prokaryotes, it is present -10 upstream, while in eukaryotes the Tata box is present -25 base pair upstream. As you can see here, -25 base pair upstream in eukaryotes, the Tata box sequences, and this is the TA and TA sequence, and then -80 upstream base pairs, when you go upstream 80 base pairs, then -80 upstream base pairs, that's the cat box. You can see cytosine, adenine, thymine box, and then -100 means that 100 base pair upstream, there is the GC box, and there is also in prokaryotes -35, or you can say 35 upstream base pair sequences. So these are the specific sequences which are recognized by the RNA polymerases, and RNA polymerase binds to this sequence, and from there then it starts the transcription, initiate the transcription, and then it follows the coding region from where these codes are copied and then carried to the translation. It finds the promoter region. The RNA polymerase starts unwinding the hydrogen bonds inside the helical structure of the DNA, and then these two strands are separated from each other. As you can see here, this is the RNA polymerase. This is the winded double helical structure of DNA. Then this RNA polymerase unwinds the helical structure by breaking the hydrogen bonds between these nucleotide bases, and these two strands are separated from each other. Once these strands are separated, then the next step proceeds, which is known as elongation. Elongation, the nucleotides base pairs bind to them at the 3' hydroxyl. In this stage, the two strands are separated. The exposed DNA bases on the antisense strand can now hydrogen bond with complementary RNA nucleotides. Now this antisense strand will complementary base pairing with the other nucleotides. Let's see here. As the RNA polymerase unwinds and opens up and separates these two strands of the DNA, the new nucleotides added up to these complementary base pairing. As you can see here, this is the strand of the messenger RNA 5' to 3'. It means that this strand is the antisense strand, or also known as the template strand. The new nucleotide adds to the 3' end of this strand of the messenger RNA of the RNA. At the end of the RNA, there is the free 3' end. So the free 3' end, so that the other nucleotide can add up to this free 3' end. And the RNA polymerase, as it adds up the nucleotide, similarly it moves forward, unwinds these helices, and similarly the new nucleotides keep on binding to this strand, and this is how the elongation of the strand occurs. RNA polymerases join adjacent messenger RNA nucleotides by forming phosphodiester bonds. So these 3' hydroxyl and 5' hydroxyl of the new upcoming nucleotide, it forms the phosphodiester bond. Nucleotides are added to the 3' end of the messenger RNA strand, which grows into 5' to 3' direction. So it will grow in the direction of 5' to 3'. RNA polymerases move along in this direction and continues to unzip the DNA while the messenger RNA is synthesized. As I already explained, that this messenger RNA, this RNA polymerases will move forward. It will unzip this DNA helices, and the new nucleotides will keep on adding on 3' on the 3' of the adjacent nucleotide, and the new nucleotide will be 5'. So 3' and 5' it will form the phosphodiester linkages, but the direction will be 5' to 3'. Let's see here the complementary base pairing for this type of, for these two helices. The complementary base pairing, as these two strands are separated, how they will form the hydrogen bonds. These form the hydrogen bonds with the complementary base pairing, as there will be adenine binds with the thymine. There will be uracil, as this is the RNA strand, you can see here. So in RNA, as always with adenine, thymine binds, but this is the strand of the RNA. So instead of the thymine, there will be uracil. As in RNA, no thymine is present, and instead of thymine, there is uracil. As you know about the rules of base pairing, adenine always binds with thymine and cytosine always bind with guanine. But here, as this is the synthesis of messenger RNA, adenine will bind with the uracil instead of the thymine, and the guanine will bind with the cytosine, just like DNA. But if you see here the hydrogen bondings, the hydrogen bond between guanine and cytosine are 3 hydrogen bonds, while the hydrogen bonds between adenine and uracil are the 2 hydrogen bonds. So this is the complementary base pairing. Through this complementary base pairing, the new nucleotides keep on adding, and this process is known as the elongation. After the elongation is completed, the sequences will come that the no more nucleotides will be added. Then this is the final step. It means that there are no more coding area, or there are no more coding regions to which the nucleotides can be further added. And this is the region which is known as termination region. The final step is the termination. As the name indicates, termination is the end. Termination is basically are the sequences which are the terminator sequences. While the elongation step, when the messenger RNA or the RNA nucleotide bases are keep on adding, when the nucleotides bases are keep on adding, and there is a synthesis of the RNA strand and it will move forward, then the termination sequences will come, then this RNA polymerase will dissociate. These termination sequences will cause the RNA polymerase dissociation, and this RNA will be set free. These termination sequences, the termination process is different for eukaryotes and prokaryotes. In case of eukaryotes, there are, as I already explained, there are termination sequences. So when these sequences reach, this RNA polymerase will dissociate and this fully synthesize this messenger RNA, or this RNA 5' to 3' will be free. As already explained, there are three different types of RNAs: messenger RNA, ribosomal RNA, and transfer RNA. In case of prokaryotes, single RNA polymerases will synthesize all these three RNases, while in case of eukaryotes, three different types of RNA polymerases will be used for the synthesis of three different types of RNAs. So when the the termination sequences reach the sequences of the terminator, some terminator sequences are more efficient at causing RNA polymerase to terminate transcription than others. So this is the most common one in eukaryotes, that there are the sequences known as the terminator sequences. When this will come, this RNA polymerase will dissociate and will set free this RNA free. The other factors are also responsible for the dissociation of RNA polymerases. These are nucleosomes. Nucleosomes may also cause the dissociation, and also there are some transcription factors that will also be responsible for the dissociation of RNA polymerase and fully synthesized RNA. While in prokaryotes, there are two methods, which is known as Rho Dependent Termination and Rho Independent Termination. In Rho Dependent Termination, when the sequences will reach for the termination region, there is a protein known as Rho protein. This Rho protein will bind to the messenger RNA and will cut it and will set it free. So this Rho dependent termination, it occurs through a Rho factor protein. This is a protein that it will bind to the messenger RNA, will dissociate the RNA polymerases and set free the RNA. But this process, it utilizes the ATP. It requires the presence of energy to perform this process. There is the other method in which sometimes these strands, these DNA sequences may form the complementary base pairing, and this complementary base pairing to the self-complementary base pairing, it will lead to the formation of the hair loop. So this hair loop is basically formed because they are rich in uracil. So when they are rich in uracil, they form the self-complementary base pairing. They form the shape of hair loop. So because of this dissociation in their shape or their structure, it will lead to the dissociation of the RNA polymerases. RNA polymerases will stop and dissociate from the DNA, and then the synthesized RNA will also be set free. All the steps for the transcription were explained in this video. Let's summarize all these steps together. First of all, what happened that RNA polymerase has to find the transcription initiation site. For this, there is a promoter region. Promoter region are the specific sequences to which this RNA polymerase will bind, and these sequences are cat box, tata box, and GC box, which makes up the promoter region. RNA polymerase will bind. Then when the RNA polymerase binds, it will start unzipping the DNA. It will break the hydrogen bonds present between the two strands of the DNA, separates them, and then when these strands are separated, followed by the elongation step, in which the new nucleotides will be added up by complementary base pairing to the template strand, and then RNA polymerase will keep on adding the nucleotides by phosphodiester bond, and the RNA polymerase will keep on unzipping and it will move forward following the elongation step. And then finally the specific sequences will come, which will lead to the dissociation of the RNA polymerases, and the fully mature and the fully synthesized RNA will be released. These sequences are known as the Terminator sequences. So this was all about the transcription. There are separate videos for transcription in prokaryotes and transcription in eukaryotes, and also for the differences between prokaryotic and eukaryotic transcription. If you want to learn in detail, don't forget to watch these videos.